the University of São Paulo,
yes,
if I remember correctly.
Yeah, yeah.
And then he went to Belgium to get his PhD. I'm not able to pronounce the name of the university in which he was.
Back then it was Limburgs Universitair Centrum.
Okay.
Nowadays they changed to an English name, so it's Hasselt University or something like that.
Okay. And now he's a professor at the University of Pernambuco, which is a federal university in the northeast of Brazil. And he just been awarded a generous grant from the Brazilian agencies to undertake his research in models of the brain and workings of the brain. And I think he's got open positions. If any of you become interested, you can talk to him after one of the talks. And it's our great pleasure to have Mauro here. And so please, go on.
Thank you very much. Thank you, André. Thank you, Carolina, for the invitation. Thank you all for being here. Like André said, I'm a physicist, or I was a physicist, I don't know. I'm becoming more and more of a neuroscientist or aspiring neuroscientist for the last 20 years or so. And I'm assuming most of you are physicists, right? That's correct, right? So I'll just assume you know nothing about neuroscience whatsoever, just nothing. And today's lecture one is going to be very peculiar in the sense that I'm not going to talk about anything I do, but I'm just going to talk about what happened in neuroscience for the last 100 years. So you have an idea of the problems, okay, what the issues are, so that in second and third lecture, I can then delve into how we can use physics to understand the brain to some extent, and to which extent, and what do I mean by that, okay? So just relax and enjoy, I hope. I'll try to paint a very impressionistic picture of this whole thing. So why is the brain so complex? That's the question I hope to have answered by the end of this lecture today. So I'll try to paint a very brief history of neuroscience. So you can start in a grandiose way, in many different ways. So I'm quoting a physicist here who claims that the human brain has 100 billion neurons, each neuron connected to 10,000 other neurons. Sitting on your shoulders is the most complicated object in the known universe. Arguably. That's a good guess, I think, for the most complicated object in the universe. For one, each of you is supposedly conscious. We have no idea how this works, okay? We have no idea how this works. It's amazing. Well, no, please don't let me talk about consciousness, okay? Because it's very tempting, and it's... So, do we have a theory for how the brain works? Or let me rephrase. Do we have a good theory of how the brain works? Well, good is a very strong word, right? Good as compared to what? So let's take the gold standard of good scientific theories. I think we all agree that our gold standard is physics. So physics has been around for a while. And I like to compare it to this golf course, which has been taken care of for a very long time, very meticulously. Details matter. You just don't go around changing this thing. It works very nicely. You go to CERN, or you go to talk to Professor Lenz in Rio de Janeiro. He's part of this collaboration, which takes antiprotons with anti-electrons, positrons. It's a hell of an experiment to bring them together, make anti-hydrogen, and measure the spectrum of anti-hydrogen to check whether it's the same as hydrogen. If it's not, there's something wrong with all the standard model. And fortunately, it's okay so far. That was the first experiment. It's the most expensive atoms in the world, anti-atoms, right? And then the second experiment, they do everything again, just in the vertical, to check whether it falls. Because it should fall. And ta-da, it does, okay? So another Nature paper. So this thing is very sophisticated, what we've been doing for the last centuries. Of course, there are some issues, like 80% of the matter of the universe, or so, according to some estimates. We have no idea what that is, dark energy, dark matter, and stuff. Okay, but it's, other than that, it's a well-polished landscape. Compared to that, to me, that's how I frame it, how I view it. Neuroscience is like going to a jungle with a knife, okay? With a huge knife, and then you cut some branches in order to move your way, and then eventually you cut a branch, you find some gold mine that nobody had seen before. So neuroscience is just full of discoveries by accident, over and over and over again. It's just fantastic. And nobody knows what's happening. You just find out stuff. And there's some poverty of theoretical understanding. And now we are in an era where data is abundant, publicly available, high-dimensional, waiting for you physicists to help, and people don't know what to do with that. So it's very it's a hot subject. So let's recap for a while. We have Newton, 17th century, late 17th century. This is when Principia came out. So the guy just invented calculus and gravitation and founded what we call science, right? And so you can choose the next landmark of your choice. I just choose Carnot's Réflexion sur la puissance motrice du feu because it's Industrial Revolution. We moved from horses to trains. That's a lot. Horses had been around for a while. And then Maxwell's grand work culminating with our understanding of what light is. People were playing with compasses and magnets and writing equations for those things, and then eventually light comes out, right? Theoretically. It's amazing. And of course, well, there was Thomson discovers the electron. Then you have Max Planck, the birth of quantum mechanics, right? You guys all study these things in your courses, in your undergrad courses. And then Einstein's Annus Mirabilis, with everything, photoelectric effect, Brownian motion, special relativity, mass energy equivalence, blah, 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 blah. And then later, general relativity. And then along the 20th century, you have the Standard Model, which is very successful. So that's physics in one slide. Just keep that in mind. So it's 3.5 centuries, more or less, old. Theoretical neuroscience, I don't know where I put the start. I think it would be generous to put it the beginning in Darwin's Origin of the Species, because it's how theoretical biology is founded upon, right? It's founded upon natural selection. And so this is 2 centuries after Newton, okay? It's 2 centuries difference. And the first Nobel Prize in physiology or medicine was given to Golgi and Ramon y Cajal, an Italian and a Spaniard, for the discovery that the brain is made of cells called neurons. I know that's trivial to you now, but this is 100 years old, okay? Only, just 100 years old. People just didn't know that. This was a mass of this thing in our brains. Someone had to look and see and convince other people that there were cells. And they got the Nobel Prize for that. Right? And then, half a century later, 63, my mom was 20 years old, was dancing, okay? So you have the Nobel Prize for Hodgkin-Huxley for discovering biophysically how a neuron spikes. I'm going to show everything in detail, in some detail, in a moment. And then people discovered how neurons talk to each other. Synaptic transmission, 1970. Brazil got the third World Cup. I was about to be born 1 year later. I'm young, okay? It's not so old. 1981, I'm choosing just random Nobel Prizes of my choice, okay? Of my choosing. Not all of them, just some that I like in neuroscience. Hubel and Wiesel got information processing, Nobel Prize for information processing in the cat's visual system. I'm going to show you a video of their experiment from the 70s. It's very interesting if you've never seen before. And Eric Kandel got the Nobel Prize in 2000. Perhaps you were born, I don't know. For relating learning to synaptic change, which is something that you might know nowadays as something trivial, but the Nobel Prize from 2000, from 25 years ago. So these things are very recent. And we haven't been able to come up with something that unifies those ideas and put those things together. So we know since 73 how one neuron works, and it's complicated, but we understand it now. But it turns out we have a bunch of them, many of them. And going from one to many, that's the whole difficulty. That's what physicists have been doing for the 20th century, right? Going from one iron atom to a ferromagnet in classical statistical mechanics, and here you are in the disorder systems school, and the whole condensed matter physics of the 20th century is about that, collective phenomenon, right? So, enjoy. Let us walk through these Nobel Prizes, one by one, slowly, so you have an idea. So, this is Santiago Ramón y Cajal, a Spanish guy who had this microscope in his home. He would observe neural tissues from different animals and different preparations, and he would make drawings with his own hands. His own drawings. This is, I forgot which animal this is. I don't know if it's a rabbit or a rat. This is the mammalian retina as measured, as drawn by him. And he couldn't publish in the journals of the time, which were all German, so he founded his own journal in Spanish, and he published there. So here's your strategy if you're having trouble publishing. This is the human cortex. And I'll now come back to this picture later today. You can see that there are different kinds of layers. So this thing is about 2 millimeters in width, okay, of the human cortex. This is the extraordinary Purkinje cell. This is one cell. This is one cell, 1 neuron of the cerebellum. So this is the neuronal body. This is where the axon goes out. And this is a dendritic tree which receives input from many, 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 many different cells. So you see that neurons are not one thing. They can come in varied shapes. They have different dynamics. And I'll come back to that later today. By the way, in order to do all those drawings, they had to stain the cells. So that's where Golgi got his Nobel Prize, together with Ramón y Cajal. And these techniques continue to evolve up to today.
How could he has these brain cells, the cerebellum human cells?
Oh, back in the day, this wasn't an issue.
Okay.
Just give me a corpse, and
Okay.
today you don't do that, right? Yeah. So this is important. This is Alan Hodgkin and Andrew Huxley. So these guys managed to understand how one neuron works just because they were happy enough that in the day, I'm showing a video now, I'm going to show you a video, they managed to find some kind of neurons which comes from the squid. So in the expression giant squid axon, what is giant is the axon, not the squid. Okay? The squid, I'm going to show you the squid. It's just a regular squid. And the axon is giant because it's like 1 millimeter thick, when a normal thing will be a couple of microns, like 1,000 times larger than a normal axon. And so they could insert wires and measure stuff. And this happened after World War II, when there was electronics in order to do that. So please appreciate what's going on historically, right? People say you shouldn't play movies in talks, but I love these movies, so I'm going to play them anyway, right? Please enjoy this movie describing what it took for them to find those equations out. Some happy accident in biology that someone found that neuron, and then the electronics of post-war development. So let's see if the audio works. Please let me know. I need a microphone.
The cephalopods' body plans and habits are so very
Sorry.
different from those of humans.
Maybe it's best if I put it on YouTube, then have subtitles to help, right? Because the audio is not so good. Please forget about it. Okay, there you go. Let's see if it works.
The cephalopods' body plans and habits are so very different from those of humans that they might almost be aliens from another world. So perhaps it's not surprising that it took a long time for scientists to discover that there are fundamental similarities between the nervous systems of cephalopods and vertebrates. Yet it was the recognition of a useful difference in their nervous system which enabled scientists to undertake research that has led to a growing understanding of the mechanisms controlling our own nervous system. The breakthrough concerned the nerves that control the contraction of the mantle muscles used in jet propulsion.
So they find out about this nerve.
As this archive film
squid.
shows, by
The squid
simultaneously contracting its mantle muscles, even a moderately sized squid can
Contract
inject a
muscles
huge
very
amount of
strongly,
water
like
with
this.
great force.
Right? So it has to be very fast and strong.
In the mid-1930s, the British zoologist Professor J.Z. Young was engaged in a study of the squid's anatomy. Young observed an array of large tubular structures, each as much as a millimeter in diameter, in the squid's mantle. As these structures were never filled with blood, they could not have been blood vessels. From their similarity to surrounding nerve fibers, Young thought they must be single neurons, giant axons, that transmitted nerve impulses from a concentration of nervous tissue, called a stellate ganglion, to the mantle muscles. Using electrodes, he stimulated
That's the electrodes
surrounding
from
nerve
the
fibers
30s.
and found that he could only produce large muscle contractions in the mantle when the large tubular structures remained intact. So, these were indeed giant axons. Scientists quickly appreciated the significance of Young's finding, for here at last was an axon large and robust enough to investigate with the techniques available at the time, and one that survived for several hours when isolated from the nucleus. The intracellular contents of the giant axon could be removed and analyzed, leading to the discovery that sodium ions were more concentrated outside the nerve cell and potassium ions more concentrated inside. By refilling the empty axons with solutions of precisely known chemical composition, experimenters were able to unravel the mechanisms of ion transport across the membrane. The giant axons are large enough and robust enough for fine electrodes to be inserted through the cell membrane and into the axoplasm. In these early techniques, a fine glass tube was first inserted into the axon and secured with thread.
You insert the glass and you tie a knot to keep things fixed.
Then the tube was used to introduce a fine wire electrode from which the voltage between the inside and the outside could be measured.
Only if you have the electronics.
But the formation of the nerve impulse was far too rapid for detailed study with any of the electrical measuring devices of the late 1930s. It
And
wasn't
then
until the 1950s, following the wartime
20 years later, you had
electronic
electronics because of
equipment,
World
such
War
as the
II.
cathode ray oscilloscope, that major progress was made. Scientists found that the nerve impulse was transmitted as a characteristic wave of electrical potential, and that this all-or-nothing action potential was generated mainly by transient movements of sodium and potassium ions across the nerve membrane. Research on the squid giant axon unraveled the mechanisms of the formation and propagation of the nerve action potential. This understanding led directly to the development of drugs that block action potential formation and so act as local anesthetics, now used routinely as painkillers in dentistry and minor surgery.
Oops. Sorry, yeah. That's the price you pay for subtitles. I couldn't download the subtitle for some reason. So, I hope you appreciate how painstaking that could have been in the 30s. It didn't work, and the 50s eventually did. And so what is the Nobel Prize worth understanding of this problem? So you have a cell, which is a neuron, which has, so the cell membrane doesn't let ions in or out, unless, of course, you have these ion channels. You have different ion channels. Some of them are specific. So let us start like this. So the neuron is a cell of the membrane. At rest, membrane potential is kind of negative. It has a little bit, just slightly more negative cells, ions inside, negative ions inside than outside. So there's a minus. So because of this looks like a capacitor, it's insulating. So they attract each other, they glue to the surface. So it's a surface phenomenon. Minus 70 millivolts. And so ion channels may allow currents to pass. And if positive current comes in, this potential becomes less negative. If it's negative current, then it becomes more negative.
Mauro, can you just come back? What are we looking at? What are the blue stuff?
This is the membrane of a neuron, of a cell.
Okay, okay.
So this is inside, this is outside.
Okay.
Okay? Yes, if you want to go the whole cell, it will be like this, but that's too complicated for us. In principle, everything I'm saying is just for the giant squid axon of Hodgkin-Huxley. I'll come back to this complication in a minute. Just imagine it's a piece of axon, yeah?
The membrane of the giant squid axon is a single bilayer?
No, of course not. No. I'm just taking one piece. This is just a model. Now this is just a model. No, yeah. No, this is just a huge simplification. I'm just talking about a piece of membrane, right? And turns out that ion channels can be specific, incredibly specific. So, for instance, you have some channels that only let potassium go through, and others that just leave sodium ions go through. Please note that they had the same valence, the same charge. It's 1. So how the hell does the channel know if it's potassium or sodium? Besides, one of them is larger than the other, right? I don't know, I forgot which is which. Doesn't matter. The one which lets the larger one go through doesn't let the smaller one go through. So yay for quantum mechanics, right? In the protein that makes up these channels. And it turns out that these channels are not, you can think of this as a capacitor, as I will show in a minute, and it's a different, this membrane potential as a potential here, and this currents going through here, like this could be some kind of resistor, right? A resistance that lets charge go from one side to the other side. It turns out that this is a resistance. it's not constant. It changes with time. Actually, it changes depending on the potential. This is something that Hodgkin-Huxley found out. So the rates at which, so potassium channels have one so-called activation variable, and sodium channels had two variables, one activation called M and one inactivation called H. And the rates at which these variables change, open and close, depend on the potential. And of course, as they open and close, sodium and potassium go in and out, therefore changing V, right? So potassium and sodium change V, and V changed the flux of potassium and sodium. So have coupled differential equations, which happens to be nonlinear. So you can systematize this thing with this kind of equation. This capacitance here refers to one side of the other side of this capacitor. You have some variable resistance here, and these reversible take into the fact that, for instance, you have more sodium outside than inside. The C is outside, okay, the cells. So if you just open a channel, sodium would like to come in. Potassium is the other way around. Potassium is more concentrated inside. So if you open a potassium channel, it will tend to go out, depending on the voltage, depending on the different concentrations, of course. So Hodgkin-Huxley managed to, by inserting that wire into the giant squid axon, block potassium channels, block sodium channels, investigate how they operate differently, and put this all together in a single equation. Well, that's actually, it's 4 equations. These things are just functions which are helping up here. So formally, it's a set of 4 coupled nonlinear ordinary differential equations. So what are these things describing? For instance, here I'm showing you what happened if you do the so-called voltage clamp equations. You go to the neuron and you hold the potential to a given level, the membrane potential, and see what the channels do. This is what they managed to do. So, for instance, these n to the power 4, so 4 activation variables for potassium, it's more or less, well, it's simpler in the sense that the more you depolarize the membrane from -6 to -40 to -20 to 0 to +20, you see that as you depolarize the membrane, potassium conductances increase. So the more it's depolarized, the more the potassium channels are open, and therefore potassium goes out of the cell. As for the sodium, which has 3 M variables and 1 H variables, I'll come back maybe later, it's more complicated. Even if you keep this depolarized, first it opens and then it closes. First it opens, then it closes. So sodium comes in and then it is forbidden to come in later on. It turns out that this is faster than this. So sodium is faster than potassium, the channels. And if you put all these things together, all those equations together, this is what you get. You get this action potential that you saw in the video, in the oscilloscope of the giant axon. So these equations give rise to this. So what's happening here is the following. Here, for some reason, you depolarize the membrane, and then sodium, the more depolarized, the more sodium comes in. The more sodium comes in, more depolarized. So it's a positive feedback. So it's very fast. And then, eventually, sodium channels start to close and potassium conductance start to increase because you're so depolarized. And then you start to repolarize because potassium goes out. Okay? And because the time scales are not exactly the same, potassium keeps coming out even after you reach the original negative membrane potential, and then therefore you hyperpolarize. And here you cannot spike again because your sodium channels are still blocked and potassium is still going out. So This happens in about 1 millisecond, which for physics, for my friends in the optics group in my physics department, it's very slow, but for us it's very fast, right? So it's 1 to 2 milliseconds. And you see what happened here is that sodium came in and potassium went out. If you do this again, more sodium in, more potassium out. This cannot go on forever. That's why you learn in high school, I think, that there is something, such a thing called as the sodium-potassium pump, which keeps doing the other way around, the reverse, in order to compensate for this, right? Slowly, in the background, spending energy for that. Right. Should I play this video, which shows everything I've just talked in video format or not? Please raise your hand if you say yes. Yes video or no video? Oh, that's a tough one. Come on, guys. No, not gonna play it. No, not.
You just said just what you said. Okay?
Yes. Yes or no? No, okay. No video. Okay, I can make it available. No, you want the video? Okay. Okay, it's just the same thing. I'll try to make it one. Yeah, it is a video. Of course it's better. Okay.
Hi everyone. Let's go over what happens to the voltage-gated ion channels during action potentials, as well as the overall ionic conductance. I have drawn a basic diagram here already of the ion channels in the membrane. This blue channel is going to be the sodium channel, while this pink one on the right is the potassium channel. The top represents the outside of the cell, and the bottom here is going to represent the inside of the cell, or the cytosol. These green flaps at the top represent the activation gates of both the sodium and potassium channels, while this little ball at the bottom represents the sodium inactivation gate. I have also drawn the corresponding graph of voltage versus time on the left. Let's start with the resting state. When no action potential is firing, both of the activation gates are closed, while the sodium channel inactivation gate is opened. No ions are flowing through these channels, and the voltage remains at roughly -70 millivolts. During this initial depolarization here caused by graded potentials, a very small portion of sodium channels would open their activation gates to prepare for the depolarization phase at threshold. At rising phase, or depolarization, all of the sodium activation gates eventually open due to the depolarizing membrane. Here, there is a positive feedback loop. The more the membrane depolarizes, the more sodium channels that open up, which causes more sodium to go into the cell and further depolarize the membrane. The sodium activation gates open very quickly, which aids in the sharp increase in membrane potential. This rapid depolarization can be seen on the voltage graph, where the voltage very quickly ramps up from threshold. The voltage goes to about +40 millivolts at the top here. At falling phase, or repolarization, the time-dependent sodium inactivation gates start to slowly close. At the same time, the potassium activation gates open. This causes potassium to start flowing out of the cell, as well as sodium to stop entering the cell. Both of these result in a rapid repolarization, where the membrane potential becomes more negative. In the refractory period, or after hyperpolarization phase, the activation gates of the sodium channels close, while the potassium activation gates remain open. This causes the slight dip below resting membrane potential seen here called the undershoot. The voltage at the bottom here is roughly negative 90 millivolts. After some time, the inactivation gates of sodium channels start to slowly open back up, while the potassium activation gates close. This allows the membrane potential to return to resting state. This step also officially resets both of these channels so that they are ready for the next action potential. The slow reopening of the sodium inactivation gates allows for the unidirectional movement of action potentials down a neuron. They inactivate the sodium channels so that they wouldn't become depolarized again. Now, let's look at the whole process altogether. At resting state, both activation gates are closed. Sodium activation gates quickly open in rising phase, and sodium ions flow into the cell. At falling phase, the sodium inactivation gates close, while the potassium activation gates open. This stops sodium ions from flowing into the cell, while allowing potassium ions to flow out of the cell. During after hyperpolarization, the sodium activation gates start to close as well. And the closing of potassium activation gates and reopening of the sodium inactivation gates brings us back to the resting state.
Okay, there you go. Enough.
Let's see what this means.
No. But that's enough. That's okay. Okay, right? So I hope you have an idea. So this is the Nobel Prize in Physiology or Medicine of 1963, right? Understanding how one neuron works. We understood. We wrote down equations for it. No. Okay. So what comes out of, yeah.
These dependencies there, M3 and 4, this was just observed. They didn't explain it from first principles, microscopic.
From molecular biology, no.
Yeah.
There was
Nothing,
such a
even
thing as
close,
molecular
yeah.
biology. They just inferred that something opened, right? And they could measure these flows somehow, these currents, and it just turned out, I don't know, maybe Silvina know the details, but they I don't know, they tried, and to the power 4, they fit, right?
Yeah.
Yes.
The use of energy in England. And so that's why Huxley ended up doing this theoretical work and mathematical work, because there was no, they couldn't use up much energy in the lab.
Oh, I didn't know
Immediately
that.
after the,
So
the wars.
mathematics is just for people
Yeah,
who
well,
cannot do the real job, like in the lab. Is that
was
it?
an interesting thing.
That's how biologists see it, okay?
But
Okay.
could they explain why is M3, I mean, heuristically?
Back in the day, no, back in the day, I think it was just heuristic, right? Just fitting the best fitting function. That have, yes, three things have to happen at the same time. So,
So it's like a cooperative effect, like it's super linear because it's like a cooperative effect, or?
yes.
Yeah, great, thank you.
Yeah, thank you, Silvina. So, I'm cheating here. She should give you the talk. This is part of the talk. So, actually, I'm going to show this in a moment, right? These deterministic equations are just an approximation, right? Because everything is microscopic. So, but if you stay here in the equations, you can, here's an eye, which is an external current you could inject in your neuron and see what happens. So you have a property here. This is from the equations, from a regular textbook in neuroscience. I think it's from Christoph Koch, I think, by physical computation. You see, if you insert a current here in the dashed line, can you see the dashed line here below? Right below the solid curve. So if you put a square current pulse here, and you depolarize just a little bit, but not enough to generate a spike, an action potential. It just goes like this. If you do it just a little bit larger, you have some kind of threshold effect in which you do generate a spike. So this is what we call excitability, in the sense that if you disturb your system a little bit, nothing happens, nothing happens, nothing happens. Eventually, boom, it happens. And when it does, it's stereotyped. It doesn't change much. How the shape of this thing doesn't change much after you do it, even if you submit it to a larger input. So in this sense, it's a kind of binary response. It's either zero, nothing, or all or nothing response. And this is how neurons communicate at distance, long distances. They use some kind of binary code, ta, 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 those all or nothing spikes.
So just a question, then if you need a given eye with a given threshold to excite the neuron,
Yes.
but if it's very big, there is no problem, or you have a problem.
Well,
I mean,
but
it has to be bigger than a threshold, but if it's doesn't matter how.
After that doesn't matter much.
Okay.
In physiological regime, of
Yeah,
course.
of course, because
In
then
the
I can
physiological
imagine
regime.
this is a problem in this system of equations. I mean,
No,
for a given value of I, well, I don't know.
Well, it can destroy your system. Yeah, but nature doesn't do that regularly. So this is the shape of the Hodgkin-Huxley spike from the equations. This is what they measured back in the day. This here is a 500 hertz signal. So this is 2 milliseconds. So you have a scale, right? So you have a scale, and this is the scale of a spike. So 1 to 2 milliseconds. That's how long it lasts. And this is for one neuron, the giant squid axon neurons. And then there's the whole rest of the universe. I'm going to comment about that just a little bit. So just to make sure we are on the same page here, the Hodgkin-Huxley model is one of the most successful models of a complex biological process that has ever been formulated. The guys started from the experimental system. They measured things. They wrote down the equations. The equations worked wonderfully, right? That's why they got the Nobel Prize. So that's one neuron. Actually, that's not one neuron. Oh, more. And suppose you insert some step current and keep it forever, like this. So if it's too small, nothing happens. If it's a little bit larger, you have one spike and then nothing. So you're just perturbing a fixed point, going back here. If you increase a little bit further, eventually these neurons start spiking periodically. So I don't know how many of you guys have taken courses on nonlinear dynamics. So what happened here is a bifurcation. So a bifurcation is a qualitative change in the behavior of a low-dimensional dynamical system. And so this is a fixed point. This is a repetitive firing. If you do the plot of the stable voltage as a function of this current, it is stable up to this point. Here the oscillations start, and therefore you have a bifurcation. In case you're curious, it's a Hopf bifurcation. In case you know what I'm talking about, it's a subcritical Hopf bifurcation. It doesn't matter. It's just a change of qualitative behavior. And if you look in a two-dimensional projection of this four-dimensional system, you have a fixed point here, and eventually you have a limit cycle, which is going like ta, 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 corresponding to repetitive firing. That will be a neuron which fires periodically. Not many neurons do that in your brain. Okay, so this is just going through the Hodgkin-Huxley equations. It turns out that this, if you've never seen this before, you might be finding it complicated. This is the simple scenario, okay? This is the most favorable scenario. You have this huge axon. You can measure everything like this. But actually, if you go into detail, people couldn't do that back in the day. Nowadays, they can insert a pipette here and probe one channel. So one channel, if you do like this, you find that its conductance is like 0 or 1. Actually, the deterministic description of the gating variables N, M, and H is actually an approximation for a very, pick up my kids at school, not going to happen. So an approximation for a very large number of ion channels. Each individual channel actually behaves stochastically with N, M, and H relating to the probability that a given channel is open, given those subunits that Silvina referred to. I'm just exposing the mass that this thing can be, okay? Besides, the Hodgkin-Huxley, like, this will be not the neuron of the giant axon, just a normal neuron in our heads, for instance. The Hodgkin-Huxley equation essentially describes what happens here, in the so-called axon hillock, and perhaps here, here, and here, and here, and here. This thing around the axon is the so-called myelin sheath, which insulates the neuron, so you don't have to spend so much energy to propagate from here to here. Everything I described here is clearly non-conservative. It spends energy, it costs you energy. Your brain consumes, I don't know, 20% of your body metabolism, even though it's only 1.5 kilograms. It must be worth something. And if you want to describe, if one wants to describe the full behavior of the neuron, the conductance-based models that some people do, I don't, but some people do, have to be adapted to each region and connected. For instance, if you want to do this describe the membrane here and here they don't have those sodium channels and potassium channels. They have other channels, perhaps. And you have to connect them, and connect them here, 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 everything together. So the dimensionality of equations is increasing, increasing, and increasing, and then eventually describe one neuron, if you want to describe one neuron in detail.
But nowadays, you would have technology to do that.
Yeah, they do.
Okay,
they do. We have close here Antônio Carlos Roque in Ribeirão Preto, specialist in doing this kind of models.
Okay.
Very detailed conductance-based models, taking into account all the known physiology of each part of each neuron that is available.
Edge equations. How
What?
close is if you take this in the
They have the same shape. So what Hodgkin-Huxley discovered was how it works in general.
Yeah.
So you have a membrane potential, it have ion channels, and they are connected to each other. Now, if you go to my brain or our brain or a rat's brain, that neuron in that specific region does have that density of ion channels, perhaps, or sodium channels or potassium channels, has other more complicated channels that people have been discovering in the last century, and they measured with a bunch of techniques and put this information together. So it can be very cumbersome, and people earn their lives like this, doing this, okay? So I don't, but some people do. And, right. It took a number of years before those guys got their Nobel Prize for synaptic transmission. So once you understand how one neuron works, you have to understand how neurons talk to each other, how they connect. And the way they connect is mostly via chemical synapses, which are illustrated here, or in a more detailed fashion here. So this is so-called presynaptic neuron, the guy who's just spiked for some reason. Ooh. Then the spikes travel to this terminal here. Once it does, the membrane depolarizes. Remember, once the membrane depolarizes, some channels open that let calcium in. Silvina here is a calcium expert. So calcium is a major signaler for many things going into the cell. So when calcium comes in here, it tells those vesicles, which are filled with neurotransmitters, to go out, dock, and do, how is it called? Exocytosis, do like this, and throw their neurotransmitter outside. Then here, this thing is close, so they are close together. This is the synapse. This is called synaptic cleft. This thing diffuses, and will eventually reach the postsynaptic neuron. This is another neuron, a different one. And then this neurotransmitter will bind to a receptor, and the effect of this binding is to open channels here, which can then affect the membrane potential of this postsynaptic neuron, either to make it more excitable, to help it fire, or to hold it down, to not let it fire, depending on the time of neurotransmitter that this guy releases here. Okay? So we have this sequence of events going on here. This takes about a few milliseconds, 10 milliseconds, or something like this. It depends on where you count the time it takes for this thing to bind and open channels here. Again, I'm not going to ask. I'm just going to play. This video is short, I promise. So this one is short. It just illustrates in cinematographic
A chemical
detail.
synapse is a special junction between two neurons that allows them to communicate without being physically connected. A synapse has several structures. The presynaptic neuron ends in a small bulb called the presynaptic terminal. The postsynaptic terminal membrane is directly adjacent, separated by a small space called the synaptic cleft. An action potential causes voltage-gated calcium ion channels to open in the presynaptic terminal.
This is calcium
The
going
influx
in.
of calcium ions prompts synaptic vesicles to release neurotransmitters via exocytosis.
Calcium tells them to release
This neuron is releasing
neurotransmitters.
acetylcholine. The neurotransmitter quickly diffuses across the synaptic cleft. Two acetylcholine molecules bind with one ligand-gated sodium channel at the postsynaptic membrane, opening the channel. When enough sodium ion channels open, the postsynaptic cell depolarizes and the action potential continues along the neuron.
There you go. Told you it was fast, right? This is just short movie. Well, there are many, many types of neurotransmitters and many, many types of synaptic receptors. Depending on the type of neurotransmitter release, a given presynaptic neuron is said to be excitatory and inhibitory. For instance, an excitatory synapse, a typical one, the classical one, releases glutamate, which is a neurotransmitter. And here you could have what is called, for instance, an AMPA receptor, which when glutamate binds to this AMPA receptor, it opens sodium channel. Sodium comes in, and then you're able to release the Hodgkin mechanism of spike generation further down the road, okay? So this is a so-called excitatory postsynaptic potential generated by this guy here. It's a small one. You need many, many neurons doing the same thing so that the postsynaptic neuron decides, okay, it's time to spike. But this is hindered by inhibitory synapses. For instance, the most abundant inhibitory neurotransmitter is called GABA, which is an acronym for something I cannot pronounce, gamma-aminobutyric acid, something like this. And when it binds to a typical inhibitory synapse, it opens chloride channels. It's a negative ion. Chloride is outside, the C is outside, NaCl is outside. So chloride comes in and further polarizes the cells. So you have a given neuron receiving inputs from excitatory and inhibitory neurons, like 10,000 neurons in total. Some 80% are excitatory, 20% are inhibitory, and this guy receives information from all those guys and then spikes or not. Every 1 millisecond, give or take. Right. So this theoretical understanding. Let me tell you one of these discoveries. This is not theoretical understanding. It's just one experimental result, not a Nobel Prize, for work developed in the late 70s by Hubel and Wiesel, how information processing works in a cat. So this is David Hubel and Thorsten Wiesel, still alive. This is not a cat, obviously, this is a human, but okay, it's the same. We're all mammals and it doesn't matter. So you have your eyes, have the optic nerve, which sends the signals to the thalamus, more specifically the lateral geniculate nucleus of the thalamus, and from the thalamus it goes to the primary visual cortex. So here you have an image, right? And this thing gets processed. There's processing in the retina itself, and then there's further processing in the thalamus, and then there's further processing in the primary visual cortex before you go to the second visual cortex, to the fourth visual cortex, etc., before you identify someone as Pablo. Okay, I could recognize him. So there's a whole bunch of processing going on. And what those guys discover is what's happening by the time the signal reaches the primary visual cortex, because it's easy to measure here. It's the back of your brain, right? So they're doing this for anesthetized cats, in which they're projecting images on the screen and measuring. What did they discover? That you insert an electrode here and measure from essentially one microcolumn, which is a bunch of neurons in a given region, and if you put the stimulus with bars like this, with different orientations, here, nothing happens. You don't measure anything, nothing. I did a little bit, more. So there's a preference for a given line orientation already in V1. So it's not detecting people or figures like this. It's detecting direction of contours in V1. And I'm going to show you a video of their experiment in the 70s, in which you're not going to see these spikes, you're going to hear these spikes, because okay, so it's a couple of milliseconds. So, and please appreciate the level of the experiments, how rudimentary they seem to us nowadays in the comfort of the 21st century like this. Back in the day, they were like this. So what you're seeing, the screen that the cat is seeing, what you're listening is the neurons spiking. There's a position. See? Spikes, no spikes. So let me record precisely. Oops. Hold on. Sorry. So this is the notion of receptive field. The neurons, that neuron, that one neuron, only responds in that position, in that orientation. I'm sorry. Some other neurons, look, respond to when you shut off the light. So there are on cells and off cells. I'm sorry that the video is playing badly. You cannot synchronize with the audio.
I don't understand what we're looking at here. That's the position of the stimulus?
You're looking at the position of the stimulus in the screen.
Ah, okay, and then?
And most importantly, the orientation of the stimulus in the screen.
Right, but I don't see the orientation changing.
No, yeah, but of course, they have to build suspense. It's,
okay.
That's...
This is only one orientation.
Yeah,
okay.
I'm going to show you in a minute, if I can manage. Let me put it. No, this is too much. This is too much. Hold on. Oh, I'm sorry. I'm messing up this thing. If I can only... I can't do this. Okay, let me... The thing is that the audio is not synchronized. If you do like this, nothing happens. So the primary visual cortex doesn't code for whole images, whole and a lot of light like this. This information has been filtered by the retina or by the thalamus. It has to have a contour. Now, for the orientation, nothing happens. Now, sophisticated techniques. See, you're blocking the receptive field of that neuron, so it doesn't fire anymore. Except for the off cells, which are on the other side. I didn't change the orientation, right? I'm sorry. This is a complex cell. Yeah.
What is the state that the cat is? Is the cat completely alive? No, he's like, I mean, the cat's alive, but I mean, not completely alive. He's like...
It's anesthetized.
Okay, okay.
The cat is anesthetized, yes.
Okay, yeah.
So there are this Heisenberg principle of sorts in biology, in the sense that, of course, you would like to test an animal in its most natural condition, as natural as possible. But if you do that, the animal won't stay quiet and will destroy your experiment, so you measure nothing.
Yeah, sure.
And so you want to measure something, you have to anesthetize the animal with some... Well, nowadays, they find ways of doing things differently, but once you anesthetize the animals, your measurements are good, but then your animal is anesthetized.
Yeah,
so you don't know whether what you're measuring applies to non-anesthetized animals or not.
Yeah.
So you have this conundrum in biology, which is there forever. It's in neuroscience, okay? So you come up with techniques like fMRI in humans. That's wonderful, because then you're alive, you're awake, and it's non-invasive, and you can see everything with a time scale of 1 second. So it's very slow. So, yeah. So here's just to show that perception is a result of activity of neurons, effectively detected, in some sense, report and represent in the brain various features. So the idea here is that you're coding for features that are being processed as the signal moves in your brain. And finally, I think I'll skip the video for this one, is Eric Kandel in learning and synaptic change. Essentially, he got the Nobel Prize for working with this animal here, which is about this size. It's a worm, it's a mollusk called Aplysia californica. And essentially, he applied shocks to these animals and showed that the animal contract some of his muscle like this, and the time of contraction was longer when you had the shock when you had not, and therefore you learned something. And then he removed the neural circuits from the animal and showed that that change in neuronal circuit had to do with synaptic changes. So when you learn something, it's because of the intensity of the synapses change with time. How does that happen? Well, this is being studied for many decades now, so people have some ideas of how that happen. There are different ways in how synapses can change. I'm going to just give you a bunch of names. So the things that, even though he worked with worms, this mechanism is common to all animals. So essentially, synaptic plasticity amounts to changes in the synapses and controls how effective two neurons communicate with each other. So you have things like short-term synaptic plasticity. So it's sub-second time scales, a rapid or down adjustment of strength with reverse to normal soon afterwards. So this is called, and you can have long-term synaptic plasticity, minutes to years. That's how you remember your kindergarten, right? Last time kindergarten stimulated your senses was a long time ago, but still remember it. So somehow it's there. And this is called, it comes by names like short-term potentiation, short-term depression, and long-term potentiation, long-term depression, and I'll have a video of how that works, but I won't play this one. So this is the state of affairs. Up to the 2000s, of course, now we are a quarter century past that. Things have evolved, but there's a need for theoretical understanding. Let me try to go back and connect a little bit with physics. I was reluctant to accept Carolina's invitation to come here, school disorder systems, because I said, well, apart from the fact that the brain is a whole mess, in the sense that it's very disordered, to some extent, to the extent that I showed you today, how can I connect with what people here more or less know or are learning, etc.? So I'll start that next lecture, but for the moment, let's talk about order and disorder in the brain, since it's a school on disordered systems. How disordered or order is the brain? Let me introduce you a very nice book by, Olaf Sporns, who's the guy who brought network science, which exploded in physics in the late 90s, early 2000s, and then he brought it to neuroscience. So I like this. This is the cover of his book, which shows you, well, a part of the human brain. And I'm going to focus on this thing. This thing is very interesting. This thing is called diffusion tensor imaging tractography, which is an MRI technique that measures water diffusion along white matter tracts. What white matter are those axons with myelin sheath that travel long distances, right? So the way it works, that you have a long axon here, right? Going from one place to another, and have water inside. So what the MRI machine does, it takes one piece here and somehow flips or interferes with the magnetization of that local region. So the water molecules here, and then those guys are going to diffuse, right? Because water is diffusing all the time. Well, in which direction it will diffuse? Well, it can diffuse here like this a little bit, but of course, diffusion will be much more significant along the axons. So they can measure that. So they okay, so in this part of the brain, the direction which molecules are more diffusing most is this direction here. In this part of the brain is this direction here. In this part of the brain is this direction here. You do this with huge precision, and then you glue those things together and build that thing here, right? This information of who is connecting to who in a large scale. So this is brain connectivity on a large scale. Which scale? Well, it's the brain. I don't know, 10 centimeters. And it's pretty much, I have to be careful, ordered in the sense that, well, your optic nerve is connected to your thalamus. And from the thalamus, information goes to your visual cortex. But there are, of course, individual variability. Not everyone is the same, exactly the same, but more or less the same, disconnecting to that happens, unless you have some neuronal condition. So what I mean is that if we recover the picture I showed from, where is it? Yeah, this is upside down. There you go, now it's the same direction. So what I mean is that your optic nerve goes to the thalamus goes to prefrontal cortex. So this must be one of these guys here, okay? Going from here, to the thalamus, and from the thalamus to here. This large, going back and forth here to left to right, is the corpus callosum, which is a major pathway connecting different hemispheres in your brain. Everybody has that. Right, so there's some order. If you look at a small scale, remember Ramon y Cajal drawings done by hand? So people with time started to identify what those things are. So this is a layer of large pyramidal cells, small pyramidal cells, some other kind of cells which turn out to be mostly inhibitory here, And 100 years after Ramon y Cajal, you managed to have this kind of picture. This is Neuroinformatics 2012, in which they reconstruct the structure of 5 so-called cortical columns, describing to this level of detail, if you want. Layer 1, 2, 3 is called sometimes one layer, single layer. Layer 4, layer 5, layer 6. And the neurons there, each column, one column like this, has about 10 to the 4th neurons. Okay? And this is 2 millimeters, this is 500 microns. It is ordered in the sense that, unless you have some neuronal conditions, all of us have these 6 layers with the same types of cells. But it's disordered in the sense that neuron 27 here connects to neuron 48 here in my brain. In your brain, perhaps, we go to the neighboring one. To borrow Carolina's comment earlier, this is not a crystal, right? By any measurement, by any means. But it's not a mess. It's not a soup. It's not a gas, right? So people have, so what is the brute force approach for modeling this? This is the Potchens-Disman paper, 2014, so 10 years old now, in which they organize, so people now know which excitatory cells, which are the triangles here, the pyramidal cells, and which inhibitory cells, subpopulations. You have 8 subpopulations, connect to which other subpopulations. So it depends on which layer you are and whether you are excitatory or inhibitory. So there's other, so relative populations and connectivity are taken from experimental data from different animals. The list of references from where they take the experimental data to construct this thing, it's about tens of references from different mammals in order to build this mapping. There's order in the sense that, for instance, there is no connection from here to here. You don't see one. There is none in no mammal. So you're forbidden to do this, to make this connection. But there's this order in the sense that this connectivity amounts, at the end of the day, in the state-of-the-art paper, to probabilities between from layer 2,3 excitatory to layer 4E excitatory is 0.008. And the number of types of neurons in those different layers are not arbitrary. There's a relative number which is conserved across mammalian species. So 20,000 of this type here, and 5,834 here, and here. And you connect these different subpopulations with probabilities, with the ones, some of them are forbidden. So I don't know whether you want to call this order or disorder, but that's what it is. Then you have to add lateral connectivity within the same layer. So there's some kind of Gaussian model to connect with some characteristic distances. And then once you connect the neurons, you have to put the model on top of each neuron. If you do the Hodgkin-Huxley, that's too expensive and not necessarily accurate, because the Hodgkin-Huxley are for the giant squid axon. So we have to adapt for each kind of neuron. So in order to avoid all this trouble, you do some simple model, like the integrated fire model, which looks linear here, but once you reach a threshold, it spikes and then resets. And this I here is the sum of all the incoming synapses according to this giant table and to those numbers. And there's also input from the thalamus, which I didn't mention here. So the thalamus sends information to layer 4. you put all these things together, you run a supercomputer, and what you get is something like this. So these are the layers. These are called raster plots. Each line is a neuron. Each dot is a spike. Ta, 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 ta. And you see that, so black is excitatory, gray is inhibitory. You measure some kind of average and standard deviation of firing rates, irregularity, level of synchrony, because this is important. And it turns out that these numbers are relatively okay when compared with experimental data of non-anesthetized animals, mammals. And you can try to run some phase diagram. You can imagine the number of parameters is just giant. They change something like relative inhibitory synaptic strength and background rate with which these things are stimulated, and you do some kind of phase diagram in which you measured AI-ness. I know that this term cannot be used like this today. AI is not artificial intelligence. This is asynchronous irregular. Okay, how asynchronous irregular activity is in a given layer, I don't know. Because this turns out to be important because this is what activity in an awake animal, in the cortex of an awake animal looks like. Asynchronous irregular. It has no apparent structure, but yet it has structure, but you don't see it. So it is called asynchronous irregular. This is a brute force approach. Putting everything together that you know, insert it in a computer, and run it. What do you get? Well, you get something, obviously. And, well, good news, that something you get looks okay. It's comparable to experiments. That's a good news. So it's a fine work. It's extremely difficult work to do. So you can go to those guys' webpage, download the model, run it yourself if you want. It's free, available, etc. By the way, the tractography I showed before, large scale, you go to the Human Connectome project, you can download the tractographies for 700 humans and run it yourself if you want. And put models on top of that. So everything's really available. And so this is the good news. It works. Well, what is the bad news? Well, depends on who you ask. If you ask me, as a physicist, my question would be, well, what exactly have we understood? I'm not sure I have understood much from what is going on. Well, I understand it's working, but why or how, to which extent, so what I intend to build in the next 2 days is now that you have an idea of how messy that thing is, how can we somehow simplify it without throwing away the baby with the bathwater by simplifying too much, right? So let's recap. Why is the brain so complex? Each neuron is itself highly complex, being mathematically described by coupled nonlinear equations in the best case scenario. There are many different types of neurons with different intrinsic behavior. Neurons interact through synapses, of which there are also many, many types. A typical neuron in the human brain, out of 10 to the 11, forms synapses with another 10 to the 4 other neurons. We're talking about 10 to the 15 synapses. Patterns of connectivity vary among different brain structures. Retina is one thing, thalamus is another thing, cortex is another thing, hippocampus is another thing, cerebellum, etc., etc., etc. Those synapses change. That's how you learn stuff. Where do we go from here? So, we'll go from here tomorrow, okay? Questions?
Mauro, does synchronization deserves a Nobel Prize? Because you did not talk about that. and it's one of the points that I have been heard several times.
Synchronization?
Yeah, synchronized ways, criticality in brains, and so on. It did not. Of course, these are communication between several neurons, and this is like a more multiple neurons interacting.
Exactly.
And, but you did not cite that. So is it somehow understood, or someone is going to have the Nobel Prize if they discover that the brain is synchronized and it has to do with any type of a function in the brain?
Yeah. So I'm not an expert in synchronization per se, but so it has received a lot of attention in neuroscience. So the idea is that in some conditions, in some brain regions, so under some experimental conditions, you do observe say, peaks in the Fourier spectrum of neuronal activity, pointing that they're working in the same region, given frequency region, right? Well, there you go. So spikes just the purest measurement of a neuron. But sometimes you insert, it's a good, I should talk about this tomorrow. If you put an electrode outside the neuron, you can measure spikes, but you can measure what's called local field potential, which is something dirtier, and as a result of many other neurons that may or may not be spiking around you. So this is easier to measure and harder to interpret, right? But you can see synchronization in this kind of signal. Sometimes you cannot do any of this because you're dealing with humans. So the first time these things appear, I think it was for humans, electroencephalogram. This is 1920s, the first electrodes. This is very simple. You put an electrode, nowadays you can buy on Amazon and connect to a meditation app, right? You can connect to a meditation app and see how your alpha waves impact your meditation quality. And just two-channel EEG put in the back of the brain, so primary visual cortex here, occipital cortex, that's very well known. You close your eyes, you don't have to be a yogi to do this. You just close your eyes, your alpha band goes up. So alpha band is around 10 hertz in electroencephalography. Well, electroencephalography is a signal that you measure outside your skull. It has to have a lot of coordinated movements, neurons, in order to be detected outside of your skull, right? Electrically. So there's a lot of synchronization going on there. So it is there to some extent in different measurements. So if a mouse is walking in a maze and you measure local field potential in the hippocampus which is a brain region which codes for memory and also codes for spatial localization, it will go to some kind of rhythmic waves that people know of. And for many times, people thought of synchronization as being the mechanism by which the binding problem could be solved. What is the binding problem? What is the binding problem? We know that I see an apple falling. The apple is red. And there's one region of my brain which codes for color. There's another region of my brain which codes for movement. There's another region of my brain which somehow represents the identity of the object. So if there's a green pear on the table and a red apple falling, and I see everything, so I'm detecting red, I'm detecting apple, I'm detecting green, I'm detecting pear, I'm detecting movement, and I'm detecting no movement. How the hell does my brain know that it is the apple that is red and not the pear, and that is apple that is falling and not the pear? If I have these different brain regions coding for different stuff, how do you, the binding in the sense of a folder that you bind together, okay, so apple, red, and falling is here, and pear, green, and not falling is here. How do we do this? The answer is we don't know, okay? We have no idea, but synchronization was thought to be a possibility because they saw this in some experiments in which they could see when you have some kind of gestalt, they somehow quantified, and they see these synchronization phenomena appearing. Almost, I don't know if it came close to a Nobel Prize, but then people realized that the stimulus was kind of stereotyped, very special. Some animals were anesthetized, etc., etc. So nowadays, it's no longer clear that it has anything to do with that. So, and there's the problem, in my opinion, that anyone that starts talking about synchronization, even in neuroscience, who do they start with? Christian Huygens. Christian Huygens, the Dutch guy who was trying to solve the major scientific problem of their time, which is how to find clocks that work in the sea so that you can know where you are on the globe, right? Because there were no clocks that worked. So they had these huge clocks like this, and the guy had his leg broken, was at home for many months, and he had two of these clocks in the wall, and he knows that after some time they were in anti-phase, tack, 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 tack. You can see many YouTube videos on that. There are infinite YouTube videos on that, right? And then I said, what the hell is going on? Then he would stop one clock and do like this and wait. And after a while, they would be in anti-phase again. Why? Because they're connected by the wall, right? So if you do mechanics, you put them, you put like a little car like this, and two pendulum like this. This one keeps the car, for momentum conservation, will keep the car at rest. If you're doing like this, the car will have to move, and there's friction. So this mode is punished by friction. Here is a car, the car is a wall, right? So that's why, what I mean by that, synchronization is very easy to attain. Two clocks on the wall. The guy had a broken leg just watching. He didn't do anything. So it could be an epiphenomenon with no particular function. It could be. Maybe from an epiphenomenon, natural selection can harness this epiphenomenon and do something about it. Could be. So it isn't clear.
So you said learning changes synapses.
Yes.
Physically, is this a change in the number of vesicles that mediate
many things. But mostly ion channels, well, it depends on which kind of the plasticity, but long term, you will have more ion channels in the postsynaptic neuron receptor, perhaps there are changes in the presynaptic neurons as well. And this goes through protein synthesis. So it's a long process. And there's more than one process. I'm just changing one. I'm just mentioning one. I'm not a specialist on that. But it's a physical one. You create ion channels. And maybe your disposition to, I don't know how much you can change your vesicle production. Does it change much? I'm not sure. I think it's more ion channels. Yeah.
So, any more questions? So let's thank Mauro again.
Thank you.
And we should be back here at 2 o'clock.